did in the lab. So use the EE mice model to look at the therapy. We use the it's novel 2AG hydrolytic enzyme. It's called alpha-beta hydrolase domain 6. So we use the inhibitor to see whether it has a protective in the EE mice model. So as we all know, cannabis or marijuana was known to have both medicinal and recreational purposes back to a few thousand years ago. However, the mechanism of the cannabis action has only begun to be elucidated in the last 20 years, thanks to the discovery of the endocannabinoid system. So the endocannabinoid system is composed of cannabinoid type 1, type 2 receptors. The ligands that bind to these receptors, the mainly is anandamide and the 2-ardonyl-glycerol, and the enzymes for their synthesis and inactivation. The two main cannabinoid receptors, the CB1 and the CB2, you can see they are both the 7-transmembrane domain G protein coupled receptors. CB1 is mostly expressed in neurons, and the CB2 is primarily expressed in immune cells or inflammatory cells. Therefore, activation of a CB1 receptor can really regulate the neuronal function, and CB2 receptor activation can control the immune function as well as the inflammatory response. So the endocannabinoid, AE and 2AG, are mostly synthesized in the postsynaptic neurons. So they are derived from their lipid precursors in the activity-dependent manner. So when they synthesized and released, like the plant-derived cannabinoid, like the THC or some synthetic CB1 agonist, they can activate CB1 receptors and cause the reduction of a neurotransmitter release. So mostly it can reduce the release of glutamate as well as GABA, depending on, so it's a vicinity where it's released. So again, to look at this metabolic pathway, so for the anandamide is mainly synthesized from N-arachidonic phosphatidyl acid, NARPE. So and then is by the enzyme is NAPE phospholipase desynthesis, and is degraded by fatty acid amide hydrolase. So this all occurred in the postsynaptic neurons. For 2-ardonyl-glycerol is synthesized from its precursor is glycerol phospholipase, and then is by the action of phospholipase C and diacylglycerol lipase to generate 2-ardonyl-glycerol. And it's a hydrolytic enzyme. It's mainly located in the presynaptic neurons. It's called monoacylglycerol lipase. So here is hydrolyzed 2AG into arachidonic acid and glycerol. Although the majority of 2AG is hydrolyzed by monoacylglycerol lipase, so it's about 85% in the brain is contribute to the MAGL hydrolysis, is contribute to 85% of 2AG hydrolysis. But certainly there are also other enzymes like alpha-beta hydrolase domain 6. It can also attribute to the 2AG hydrolysis, but this occurred in the postsynaptic neurons. And they can also be oxidized by COS2. So even they contribute a small portion of 2AG hydrolysis, but because they are different cell localization, so indicating this enzyme might regulate the 2AG hydrolysis in a kind of cell type, probably in a cell type specific manner. So that also indicates they probably control the different pools of 2AG hydrolysis. In fact, if we inhibit both MAGL and ABSG6, you can see the additive effect of the 2AG hydrolysis. This is demonstrated by Nephi group. So suggesting that these two are really contribute to different pools, and so we regulate the 2AG hydrolysis kind of in the different cellular components. So the activation of microglia, because in the microglia they also have cannabinoid receptor expression, like CB2 receptor. So the 2AG or AEA can also activate the CB2 receptors in microglia cells, and then they cause inhibition of the inflammatory molecules release, like cytokines, nitric oxide, and IOS. Therefore, they can prevent neuronal toxicity. So in compared to the plant-derived or synthetic cannabinoids, the targeting endogenous cannabinoids has the following advantages. So it can enhance AEA and 2-AG synthesis because not like the other neurotransmitters, they are kind of stored in the synaptic vesicles. So this AE and 2-AG are synthesized on demand. So that means they only synthesize when and where they are needed. So therefore, the inhibition of its hydrolytic enzyme can activate cannabinoid receptor locally rather than globally. So if you just use the general agonist, because they can activate CB2 receptors expressed in every tissues. Enhancement of their levels can avoid psychotropic effect. So because this can avoid the psychotropic side effect caused by CB1 receptor activation. From this metabolism, we can say if we inhibit this hydrolysis, the AE hydrolysis, as well as 2-AG hydrolysis, it's not only increase the endocannabinoid levels, but it can also reduce the arachidonic acid release and the subsequent prostaglandin signaling. So that means, so this endogenous use kind of inhibitors of this enzyme can enhance both endocannabinoid signaling and reduce the eicosanoid signaling. So there are lots of studies and use these inhibitors as well as knockout mice to see they have therapeutic effect, like in the inflammatory and the neuropathic pain, and epilepsy seizures, and stroke, TBI, and lots of neurodegenerative disease, Alzheimer's, Parkinson, Huntington, and ALS, as well as lots of studies in the multiple sclerosis. However, so if there's currently their study use the chronic inhibition of monoacid glycerolipase, as well as knockout mice, they found there's boost of 2-AG production endogenous. So the enhancement of 2-AG can also desensitize the cannabinoid receptors. Therefore, they can cause the behavior tolerance. So that means if you increase the 2-AG level in a much higher level, they can cause some side effect. So that study also kind of indicate if partial inhibition of 2-AG might be more therapeutically useful. So regarding the ABSD6, because ABSD6 only contributes to a small portion of 2-AG hydrolysis, so it is hypothesized that if we inhibit this enzyme, might increase the 2-AG level in the therapeutic window without causing the psychotropic, without causing the CB1 receptor desensitization and some psychotropic side effects. In fact, in the last 2 years, there's a few studies to look at the ABS6 in the animal model. So in the RPS-induced inflammation model in mice, and when you use the ABS6 inhibitor, certainly it can block the inflammatory response. So it's mostly kind of acting on the macrophages. So also in the epileptic seizures, so in Nephi group, they found so the ABS6 inhibitor has a very good anti-epileptic effect. I mean, certainly, so the cannabinoid in the epilepsy field is basically and has been demonstrated in several kind of human patients, and certainly they are very responsive to the cannabinoid agonist. So in our group, we also demonstrate the inhibition of ABS6 has a very clear anti-neuroinflammatory and neurodegenerative effect, and certainly these inhibitors can also shift the microglial macrophage from M1 to M2 phase. So we thought so from the anti-inflammatory, from the pro-inflammatory to the anti-inflammatory, and because they have both anti-inflammatory and neuroprotective effect, so we are thinking so these inhibitors might be also good for the multiple sclerosis. So use a marker-induced EEG mouse model. So we treated the animals at day 10. That's basically immediately on the disease onset with these inhibitors, and with 10 milligram per kilogram. And then once a day until the end of experiments, you can see the behavior scores, the clinical scores was significantly inhibited. So the blue line is WLL treatment. So if we co-administer with the CB2 receptor antagonist, so we can see the clinical scores get increased. So, but in the CB1 antagonist, so there's no change, indicating this drug effect is mediated by CB1, CB2 receptor activation. And because we do the drug injection in the IP, so we don't know so where it works. So there's we use the 3 different methods to see the drug is really have the right target. The first method we use is called activity-based protein profiling. So it's the novel proteomic technology is utilize the active site probes and then can profile the enzymes of its enzymatic activity in the proteome. So basically, if the enzyme is bound to its inhibitor, so the probe will not bind to the enzyme, right? So, but it's active enzymes, they can bind to it. So in that case, if we do the in-gel scanning, so when it's bound to the inhibitors, we will not see the band. So this can see the selectivity and and as well as the potency of the drug. So how is it inhibited in vivo, especially in the central nervous system. So here is the results. We found it, so when we treat WWL, we see significant inhibition. So it's also not completely inhibited. So it's about 50% inhibition. But in the vehicle and certainly in the control animals, you can see clearly the band of ABH6. And we also look at the 2-AG hydrolysis use radioactive 2-AG. And we can see in the control animals, the WWL treatment only reduced 4% of 2-AG hydrolysis. So that's consistent with the report in the brain. So the ABH6 only contribute to 4%. So, but in the EE animals, so the inhibition comes to 12%. That means there's 3-fold increase of the 2-AG hydrolysis contribute to ABH6. So that's indicating ABH6 dependent 2-AG hydrolysis certainly increased. And also we look at the 2-AG levels, use mass spectrophotometry. So use the WWL's 10 milligram per kilogram can cause 2-fold increase in the brain as well as in the spinal cord. So that's clearly says that the inhibition of ABH6 can increase the brain levels to 2-AG. And then we look at the T-cell infiltration and the microglia activation. You can clearly see the WWL inhibited and is blocked by CB2 antagonist, but not the CB1 antagonist. So consistently, there are several inflammatory markers like COX-2, TNF-alpha, and INOS, as well as some other cytokines. We can also see this inhibited, and also it's CB2 receptor dependent phenomenon. So these changes might be correlated with NF-κB phosphorylation. So because we also see there's significant phosphorylation of NF-κB. And also we use the in vitro study in the microglia cells. We see the microglia is also significantly increased, all these inflammatory markers, also in the CB2 receptor dependent manner. So we also look at the active leukocyte cell adhesion molecule. That's basically is one of the indicators of BBB. So when the increase indicating there's BBB increase. So you saw the blood-brain barrier is also getting inhibited. So the next question is we ask, so which immune cells, because we found the ABH6 is mostly expressed in the immune cells. So which immune cells kind of express the ABH6? And we did confocal, and we see the ABH6 is mostly co-localized in the microglia, but not in the T cells. So this consistent with in BV2 cells, so ABH6, but not monoacid growth lipase is expressed in the microglia cells, is contributed to 2-AG hydrolysis. Also consider the CB2 receptor is mostly expressed in the microglia cells. So we are thinking, so increase ABH6, so probably will increase the 2-AG hydrolysis in the microglia cells. If we inhibit it, will block the microglia activation. So this we look at several other kind of markers. So this basically CC1 is oligodendrocyte marker, is mature oligodendrocytes. You can also see the kind of reduced. And we use the LOX phospho-blue and to look at the myelination, and the silver staining to look at the axonal injury. You can clearly see in the EE they have kind of get injured, and so in the treatment, certainly they completely recovered. And this phenomenon also mediated by the CB2 receptor rather than CB1 receptor. So the next question we ask, so if this is CB2 mediated, so if we use CB2 knockout, so certainly the WR will not have a therapeutic effect if it's only mediated by the CB2 receptor. In fact, this is the case. So this, again, so this is the Y type, and the WL70 have clearly kind of inhibitory effect. So in the knockout, so if you can see here, so there's a little enhancement in the clinical scores. And when we treat it with WL70 in this knockout, they do not have any effect. And also true if we look at the microglia division, so certainly in the knockout, the WL70 failed to have any protective effect, as well as in the myelination stand by MVP and axonal injury like IMF200. So in this immunohistochemistry, we also didn't see any, the new axonal degeneration and the demyelination also is not protected in this CB2 knockout. They're clearly indicating this inhibitor is through the CB2 receptor activation. But you probably also notice, so at the beginning of the talk, so when inhibited all these enzymes, it's not only enhance the cannabinoid signaling, it's also reduce the eicosanoid signaling. So then we also look at the prostaglandin production, and clearly in the EAE, so there's the enhancement of PGE2 production, and it's inhibited by WR70. Again, in the microglia cells, we can also see so the same kind of phenomenon. So the 2AG the prostaglandin increase in the LPS-treated cells, and because the 2AG is also a substrate for this prostaglandin production. If we add 2AG in the presence of LPS, they see a huge enhancement of the PGE2 production. This enhancement also blocked by WR70, so that's indicating in microglia cells. So this really 2AG is the substrate of, arachidonic acid is by the action of ABSD6. So at the end, we think the WR70 have kind of two, basically the mechanism is through increase the 2AG signaling as well as reduce the arachidonic acid release and the subsequent prostaglandin signaling. So in conclusion, so certainly they can ameliorate clinical scores and suppress inflammation and reduce the loss of mature oligodendrocytes, and all this effect is mediated by CB2 and not CB1. So clearly this effect, WRS effect is in CB2 receptor knockout mice is kind of get lost. And also the effect might be contribute to the prostaglandin reduction by this drug. So here is basically the people who did the work, and thank you. Yeah.
So if you start treating mice maybe later instead of on day 10, but after the clinical disease, so in that model, like the clinical disease comes up and somewhat monophasic. And so during that part, if you treat, can you drive the disease down, or do you think it's too late?